testing, test. Okay, so let me check my mic. I think my mic is okay now. Okay. Okay, I think my mic is okay now. So now it's already 8:01. Okay, so 8 o'clock. So we will start our class now. Okay, so for today we will enter new chapter, which is the last two chapter for Sem 2 lah. Chapter 12, immunity. So yesterday I already finished up the chapter homeostasis. So hopefully you guys understand it. For those who attend lah. Okay, so for today we will enter the new chapter, chapter 12, immunity. So there are 4 topics that you need to learn: immune system, development immunity, concept self and non-self, and finally is autoimmune disease. Okay, so this chapter I can say is totally new chapter to you guys. You never learned it before in SPM. I can say this. So this chapter I can say is one of the harder chapter, because you never learn in SPM. You does not have any basic in SPM. So in this chapter will be more hard if you want to learn it lah, especially if you want to understand it compared to previous chapter. Because previous chapter, although you have learned new things, but actually you have the basic concept in SPM. Like the last chapter, homeostasis, the formation of UV, although there are many new things, but actually you already learned in SPM 2. You have already have a general image on that. But in this chapter, I can say totally new to you. And for me, I think it is more interest topic lah, more interest chapter, because in this chapter you will learn about the immune response, the immunity response toward of the human, how our body, the immune cell, the immune response protect our body against the pathogen. So for one of the example like COVID-19 lah, right? If you get the sick or get COVID-19, why some people can secure? Why some people can get well soon, but some people will die, will get immediately, need to have additional treatment. So it is based on the immune response of you. So every people will have this kind of immune response. Okay, so for this topic, hope that everyone need to alert it lah, because it is totally new to you guys. Okay, so I just go through the basic overview of the immunity that you learn. Immunity. So the definition of immunity is very simple. It is defined as the resistance to disease, specifically infection disease. Just resistant to disease only. It want to prevent your body to get that kind of disease, to prevent you sick, to prevent from the sick. I want to maintain your healthiness. Okay, so I just make a recap that you learn in SPM. If you remember in your SPM that you learn about the immune response, I think got certain chapter you learn about immune response in SPM 2. In SPM, you know that you learn about the body defense system. There are three level of body defense system, right? First line defense, second line, and also third line, right? That you learn in SPM. And in SPM, normally you are focusing on first line and also second line only, right? Innate immune response, which we call as first line, and second line we call as innate immune response. So we know that first line is just a skin, like the skin barrier or your epithelial tissue that protect the microbe, protect the pathogen or any harmful substance enter into your body. But once the microbe enter your body, then it will trigger second line, right? So second line, the main function is phagocytosis, right? phagocytosis will occur. Your red blood cell, some of the red blood cell like macrophage, dendritic cell, natural killer cell, and also neutrophil, basophil, all these leukocyte will be stimulated and will trigger phagocytosis, right, to engulf all the pathogen. This is second line. And then in third line is the most important that you will learn in our syllabus. Because in third line, I think in SPM you only learn about antibody, right? You only learn about antibody interaction, and then you only learn about the artificial acquired natural passive immunity or natural active immunity, or artificial, right? Artificial active and artificial passive. All these, right, that you learn in SPM. So in STPM you will not learn that, but you will learn about the more detail of the third line defense system, which is we call as adaptive immune response, which is more detailed. If the pathogen already skip first line, okay, it already enter the cell, and then after that they enter the cell, still the leukocyte cannot settle it, cannot engulf all the bacteria, all the pathogens. So third line defense will be stimulated. It was starting to stimulate and starting to do protect your body. So third line defense, what we call another name, more scientific name, is we call as adaptive immune response, will develop more slowly and mediate later, even more effective and highly specific in defense against infection. Yeah, so compared to first line and second line that you learn in SPM, it is not specific, unspecific, low specific. It's just that all the leukocyte, especially second line, all the leukocyte like neutrophil, basophil, macrophage, dendritic cells that you learn in semester 1 about the type of the blood cell that you learn, all that except lymphocyte. Okay, all these leukocyte will trigger phagocytosis and to engulf all the bacteria in the second line. But then the second line is still not enough, then third line will be induced. Yeah, so our syllabus, we will more focus on third line defense. Okay, so in third line defense, we will more focus about the lymphatic system. So in SPM, as you guys know, in SPM, the function that you learn, that you know in lymphatic system is to provide immune response. Lymphatic system is main system to provide the immune response to us, right? So you know that lymphatic system is also one type of circulation transport system, formation of interstitial fluid, like that you learn in chapter 8. So lymphatic system also provide the immune response to. Okay, so in lymphatic system that you need to learn is lymphoid organ. So there are many lymphoid organ that you need to know. So you can see here this diagram: bone marrow, thymus, spleen, tonsil, adrenal, lymph node. All these are considered as lymphoid organs. Especially bone marrow, spleen, thymus, and lymph node. These four are very important. And also, these four are very important lymphoid organs. Not say that others are not important, but these four are very crucial in play the role in adaptive immune response. Okay, so we will see it one by one. Okay, bone marrow. Okay, I think what are the bone marrow? I think I don't need to say, right? So bone marrow is you can say it is one type of the lymphoid organ that supports self-renewal and differentiation of hematopoietic stem cell into major blood cells. So it is the site of the production of all the blood cells. So all the blood cells will be produced in the bone marrow. Why? Because bone marrow containing the blood stem cell, we call as hematopoietic, HSCS. This is the stem cells in the bone marrow where it can it can develop and grow, become the major blood cell. So all the blood cell that you learn in semester 1, all the blood cell, erythrocyte, leukocyte, and also platelet. And then we know that leukocyte ada banyak kan. You got basophil, you got neutrophil, you got lymphocyte, basophil, neutrophil, lymphocyte, macrophage, dendritic cell, natural killer cell. All these are considered as leukocyte, right? So all of these blood cells are come from this stem cell. So bone marrow are very important. If your bone marrow is unfunctional or useless, then no blood can be produced. Your formation of blood cell will have a problem. Okay, so lymphocyte play the role in the adaptive immune response will be produced from bone marrow. The most important in third line defense, lymphocyte play the major role. Other type of leukocyte like neutrophil, basophil, macrophage, all are macrophage except macrophage. Basophil, neutrophil, and eosinophil, and dendritic cells and natural killer cell. All these are more focused on second immune response. They focus on second immune response. In third line immune response, lymphocyte play the role. And then bone marrow is the place where the B cell mature and your T cell will mature in thymus. So thymus is another type of bone marrow. So if you still remember that our lymphocyte containing two main of lymphocyte, right? Either are B lymphocyte or T lymphocyte. So how to differentiate then? Because if you see B and T, already the naming of them, B already mentioned bone marrow because it is synthesized in bone marrow, growth in bone marrow, and also mature in bone marrow too. But the T cell is synthesized in bone marrow, but later it will move to the thymus for mature. So the maturation of T cell is at thymus. Why it is T cell? Because of thymus. T refer to thymus. So that is why they're naming as B and T. B is because of bone marrow, right? B. And then T cell is because of thymus, T. Okay. And then there are also these are just characteristic cells on the bone marrow they contain. So it is one type of the bone marrow because I already say bone marrow bone. So it also containing osteoblast, basic unit of bone cells that you also learn in semester 1 about the connective tissue, right? Bone cell. So endothelial cell, reticular cell, and also sympathetic neuron. There are also neuron at here. Okay, so this is just function of their osteoblast is the cell that both generate bone and also control the differentiation of your stem cell. And endothelial cell is the blood vessel lining wall. And then reticular cell are same process connecting cell to bone and blood vessel. And another one, sympathetic neuron, is to release the stem cell from the bone marrow. So this is the general structure of the bone marrow. So you can see lah. Okay, another one, thymus. This is another organ I already mentioned. It play the major role in maturation of your T lymphocyte. So after your T lymphocyte is synthesized in bone marrow, after that they will move to the thymus for further maturation. So this is the most important part for thymus is lymphoid organ 2 in the thoracic cavity above the heart. The location of the thymus is actually above the heart only. So you can see this is your heart, right? So above your heart, actually the outer surface they containing thymus. Okay, so the remaining part is the explanation of your thymus function. So normally T cell precursor will still retain the ability to give rise in multiple homeostatic cell type travel by the blood from the bone marrow to the thymus for maturation. This is just about how the T cell transfer to the bone from bone marrow to thymus. So thymus containing the specialized environment where your immature T cells can generate unique antigen receptor we call as E cell receptor. This one later I will explain what is this T cell receptor which play major role. T cell receptor ni play major role in the immune response. This one that you will learn more detail in development of immunity. But you need to know that T cell and B cell lymphocyte, they containing T cell receptor and B cell receptor, and these receptor are very important in adaptive immune response. Okay. So this receptor receptor will be produced and then be selected based on their reactivity of cell MHC peptide complex expressed on the surface of thymoid stomach cell. Another terms we call as MHC protein major histocomposite complex, the full name. Okay, MHC. So this one is another terms that is very important too during the development immunity. Many terms so you need to learn. But for this one, I just want to tell you that it is the maturation process of your T cell. Okay, so this T turbocyte will bind self MHC peptide with an intermediate affinity, undergo positive selection, resulting their survival, maturation, and migration to thymic medulla. Okay, for this one, I just want to explain you only. This is very simple only. Okay, thymus, we want to produce the T cell receptor. And then this T cell receptor is very important. They need to bind to the antigen. They need to trigger you need to trigger the action of your T cell. You need T cell. So your T cell receptor need to bind to the antigen first. So for this maturation process, T cell receptor is only the receptor. We only want to produce the receptor that only bind to the outside pathogen, the harmful antigen only. The self antigen that inside our cell, meaning outside our cell, is not will be detected by our T cell one. So this maturation process will detect, select only the T cell receptor that can react with the harmful antigen only. The self antigen will not be detected. So it only select the harmful antigen receptor only. So the if the receptor that can bind to the self antigen, so that T cell will be die. That T cell is we don't want. The body don't want that kind of T cell, because this T cell will attack our body. So we don't want that type of T cell. So we only want the T cell that can bind to the harmful antigen only, not bind to the self antigen. So this is the general process I need to tell you, the sentences that provide these two. Okay, I hope clear. Another one, lymph node. Lymph node, I think these terms everyone hear it many times. I think in SPM you also learn, and then you know that lymph node, the general function play the role in immune response. It containing many leukocyte, many white blood cell to trigger immune response that you learn in SPM, right? But actually lymph node is very complex, and you can say it is very complex, and also many adaptive immune response occur at there. So you can see this diagram. So this diagram is the general structure of your lymph node. So you can see here in lymph node they containing two zones, which we call as T cell zone, paracortex, and also follicle zone, B cell zone. Meaning that all your T cells will be accumulated at here, and all your B cell will accumulate at here. And then this is your antigen presenting cell, and this is your antigen presenting cell, and this is your lymph. So you know that lymph node is one type of the bean-shaped organ, right? If you see the general structure of your lymphatic system, you can see here. Bulat ni. You can see there. Circular shape, bean shape like this. All these are lymph node. So actually in our body we are exposed and surround many thousands of lymph node in our body, especially at here, at our neck. In our neck, in our neck, and also at our shoulder, neck and shoulder, and the middle part, and also our sexual organ, near the sexual organ, containing rapidly number of lymph nodes. All the lymph nodes will accumulate at here. Okay. So lymph node, as we mentioned before, it play the role in adaptive immune response. Yeah. The major role if lymph node is regulating your adaptive immune response to activate your B cell and T cell. So in general, because you are not learning yet the immunity development, later I will teach you. In general, for now you just want to know that your lymph node play the role in adaptive immune response for now, because you need to activate B cell and T cell. Because as I mentioned before, third line defense, adaptive immune response are highly specific. So what is the meaning of highly specific? You will still remember enzyme. You know that enzyme is highly specific. What does it mean? Meaning that if A enzyme, it only can catalyze A substrate only, right? So like maltose enzyme. Maltase enzyme only can catalyze maltose only, right? Can maltase enzyme catalyze amylase? Cannot. If can maltase enzyme catalyze fructose? Cannot. Not fructose. Galactose. Can maltase enzyme catalyze galactose? Cannot. Maltase only can catalyze maltose only. So that is why that we call as enzyme is highly specific. Similar to this, your adaptive immune response, your B cell and T cell are also highly specific. Only can trigger or bind to certain specific antigen only. A antigen is A antigen, like COVID-19. Because we know that there are many variants of COVID-19, right? So normally your cell is only can trigger like the alpha COVID, right? Maybe got alpha or what variant? Delta. One of the most famous one, Delta COVID. So Delta variant. So if your immune response already trigger Delta, so Delta activate your trigger. How to say? Immune response already activated to against the Delta variant, so that your immune response only can fight against the Delta COVID-19 only, but others they cannot. Cannot, because others maybe have other changes. So it is very highly specific. Only can respond to certain specific antigen only. So this is just I want to tell you. So later you will see it more clearly when you learn about the development immunity. Okay, so lymph node provide your ideal microenvironment to encounter between antigen and lymphocyte and productive organized cellular and humoral immune response. So just that it provide the environment because it provide the environment between the antigen and lymphocyte, so that you will see that in lymph node there are T cell zone and also B cell zone, right? And then there are lymph. So your antigen will flow at here. All your antigen, all your leukocyte will gather at here. Akan berkumpul di sini. And then starting to fight. So your B cell and T cell will starting to activate and then to fight against activate your B cell, so that once the B cell and T cell are activated, so your lymphocyte or this activated B cell and T cell will go to other part to play the role of immune response. So the main function of your lymph node is to activate B cell and T cell. Not necessarily say that lymph node are the place only play the role in adaptive immune response. So actually adaptive immune response can play everywhere. Once they are containing activated B cell and T cell. So if your B cell and T cell is already mature but not activate again, you don't give the task to them like orang biasa kan. B cell and T cell like orang dewasa sekarang dia dah jadi orang dewasa, tapi dia tak pergi kerja. Kalau dia tak pergi kerja, macam mana dia nak tahu saya nak buat apa? Sekarang dia tak tahu nak buat apa. So you need to give the task to B cell and T cell. So lymph node is the workplace for B cell and T cell. So sekarang dia, and once the B cell enter to the follicle, then you know, apa saya kena buat? Saya kena buat ini. T cell saya pun tahu, saya kena buat ini. Then dia tak tahu, lepas dia keluar, then pergi, dia tengok ada apa antigen yang saya tahu, dia terus fight dengan dia. Macam tu lah. Lymph node macam itu satu tempat untuk gather information punya, dapatkan informasi tu untuk tahu musuh dia. So inilah tempat dia. Okay. So already mentioned cortex contain B cell lah. So normally is B cell containing B cell, and also not only B cell, macrophage, dendritic cell also inside at the cortex. Okay, another paracortex containing T cells, so and also dendritic cells, some of the dendritic cells. And then medulla is the innermost layer, and the side lymphocyte acid and the lymph node through the efferent lymphatic. Okay. Ada lagi. Okay, and then this one is more about the immune response. Okay, so your antigen will travel from the infected tissue to the cortex of the lymph node by afferent lymphatic vessel. So all these, you can see these at here, afferent lymphatic vessel. So all your antigen will travel at here. Dia akan bawa your fluid, your lymph will travel, will take all these antigen and travel them to the lymph node. Okay, so once your antigen is already enter the lymph node, they will enter either in particular form or present or is processed and presented as peptide on the surface of migrating antigen presenting cell. So okay, so this one I will explain in more detail during the development immunity, but you need to know this function where the antigen presenting cell will maybe occur at here. And then particular antigen will be trapped by the single antigen presenting cell, and it can be passed to other antigen presenting cell. Okay, so alternately, particular antigen can be processed and presented as peptide adhesive complex on cell surface, so that already in the T cell rich paracortex. Okay, so for this one, actually is the general process about the development immunity lah. So this one I hold on first. If I hold on first because it's very complicated process, I will explain in more detail when I reach the development immunity. But you need to know that adaptive immune response, the initial step of your adaptive immune response, you need to activate B cell and T cell. Activation of B cell and T cell will need to occur at lymph node. Okay, the main function you need to know. Okay, and then this is the general process, but general process. Okay, another organ, spleen. Okay, spleen is play the major role in mounting immune response to antigen in the bloodstream. Okay, so normally spleen is situated in the high in the left side of your abdominal cavity. Is a large ovoid secondary lymphoid organ. Okay, there is one type of lymphoid organ. So the major is mounting immune response to antigen in the bloodstream. Okay, so apa maksud dia? Okay. So if you compare to lymph node, what are the difference between spleen and lymph node? Lymph node are specialized for encounter between lymphocyte and antigen drained from your local tissue, but your spleen is specialized in filter blood and trapping blood-borne antigen. Faham apa itu? Maksudnya lymph node tak kisah, dia is specialized for encounter between lymphocyte and antigen. So tak kisah apa antigen, apa lymphocyte, dia masuk je, kerana dia nak activate your lymphocyte. So apa sahaja antigen dia masuk je. But this one, it filter the blood. and trapping blood-borne antigen. So meaning that only antigen can be passed through. The blood vessel, your lymphocyte, blood cell cannot pass through into it. So you will see this diagram. So this diagram is your blood vessel, right? Splenic artery, and splenic vein. And then you will see this diagram, or you will see this. There are the white part, and also red part at here. Okay. So hence your spleen is particularly important in the response to systemic infection. Okay, so blood-borne antigen and lymphocyte are carried into the spleen through the splenic artery and out from the splenic vein. So spleen is surrounded by a capsule from which a number of projections called as trabecular extend, providing the structural support. Okay, so there are red part. Red part consists of sinusoid populated by red blood cell, macrophage, and some lymphocytes. So it is the site where all and defective red blood cells are destroyed and removed. Yeah. So you see this diagram. So this red part at here. Ini semua ni. White part is at here. Yang ni brown, this yellow color and also brown color. And then others one you can see yang outside ini semua we call as red part. Okay, so red part is a place to destroy all erythrocyte. All red blood cell will be destroyed at there. And then white part is the place consists of the PALS, peripherally lymphoid sheath cell, populated by these lymphocytes as well as B cell lymphocyte. So this play major role to specialize macrophage and B cell and first line defense against the certain blood-borne pathogen. Okay, so you need to know that spleen is act as the melting process. It filter only. No activation process here compared to lymph node. No activation. It's just filtering the blood and trap the blood-borne antigen only. Only the certain blood will be passed through only. Only the certain antigen can be filtered into it only. Not all the antigen. Compared to lymph node, semua antigen, all the antigen will be go into that. But for spleen, only certain antigen, only the blood-borne antigen can enter into it. Okay, so this is about the spleen. And another one is about you can see there are tonsil appendix, right? So this is the lymphoid organ which we call as mucosa associated lymphoid tissue, MALT. So actually this is a lymphoid tissue. This is not difference between the previous one. The previous one is one type of organ. They are totally an organ, but this one is because of tissue. So it is located at random places. There are no certain, not like an organ like in a certain part play their function. It is like many tissue gathered at here and they play many function like that only. Okay, so you see this diagram. So this diagram is one of the example about the about your this is one of the diagram of your MALT, which the payer page. So payer page. So this one. So there are three lah. So one of the example I give you is payer page. Okay. So this actually is located at your located at the cells. Okay, so the structure of GALT is well described and range from loose barely organized cluster in the lamina propria of intestine wall to well organized structure. So normally this payer page tissue, so you can see there these all are payer page tissue. So actually this one is the structure of your small intestine, located at the small intestine. So your overall body function is underscore by each large population of antibody producing plasma cell, so that your number exceeds of your plasma cell in the spleen, lymph node and bone marrow combined. So this is just play the role of combination to increase the production of your lymphocyte only, like that production of lymphocyte so that it can trigger the adaptive immune response more easily. Okay, so that's all about the lymphoid organ. Okay, so this lymphoid organ are very important too, because this is a basic theory that you need to learn before you know about the development immunity. So you need to know the location of the occur in the development immunity. So you know that activation is occur at the lymph node. Your activation of your B cell and T cell is in the lymph node. Okay. Now we go to the development of B cell and T cell. Okay. So I think this one, I don't know MPM consider this topic or not. But if you refer to MPM syllabus, they say one word learning outcome, explain development of B cell and T cell, and then you refer to the books of the like Campbell or any Oxford. I think Oxford does not mention about the development of B cell and T cell as well, and even in Campbell does not, because this actually is very complicated process. Development of B cell and T cell is very complicated process. So I don't know why MPM want you to learn about this, explain, because you want to explain. So I cannot, so I have no choice, so I need to explain to you guys. So this process I can say is very complicated. I hope that MPM tak keluar lah. So I hope that MPM tak keluar lah. Okay, so in general, I just want to show you the general that you learn in Sem 1 about the blood cell, type of blood cell. So we know that I already mentioned about stem cell. This is our stem cell. So it will differentiate, develop into many white blood cells, many leukocytes. So the most important I need to focus is here, lymphocyte. So lymphocyte. So we know that we containing two kinds of lymphocyte, T lymphocyte and also B lymphocyte. So T lymphocyte later they will develop, either there are two subtype of T lymphocyte, helper T cell or cytotoxic cell. Or if you refer Oxford, they say T4 and also T8. Yeah, ada beza. You need to alert helper T cell and also cytotoxic T cell, and another one B lymphocyte you develop into B cell. So B lymphocyte, helper T cell, helper T lymphocyte, and also cytotoxic T lymphocyte. Okay, so in development you need to know how this cell can be developed sampai sini. Macam mana in the process, this process. So yeah, you need to learn. So I just make it short, the development of your lymphocyte. So how the T cell and B cell be developed. So you know that both of them are lymphocyte, but how to differentiate? Macam mana kami boleh tahu ini T, macam mana boleh kami tahu ini B? So I already mentioned, you see the maturation location. The maturation location, or if the lymphocyte is matured in thymus, so it is T lymphocyte. If maturation process in bone marrow, then it is B lymphocyte. And then both of them are synthesized in bone marrow. So this one I just repeat again what I say before. Okay, so I think I don't need to talk more lah. Okay. Okay, next I go to development of T cell. Okay, so if you still remember, T cell containing two types, right? Helper T cell and also cytotoxic T cell. Okay, so how to differentiate them? Kenapa ada dua ni ya? How to differentiate them? Helper T cell. For now you don't need to know the function first. You just want to know how to differentiate them. Kenapa ada dua ni ya? How to differentiate apa dua ni? So okay, so later your CLP, which is the common lymphoid progenitor. So this is the precursor of your T cell, will enter your thymus from bone marrow, and then so it will express your maturation lah. So no antigen receptor yet. So the immature T cell now is known as thymocyte, will produce a functional, safe, and useful T cell repertoire in the thymus. So now your T cell is known as thymocyte, which does not contain any T cell receptor yet. No T cell receptor yet. Okay, after that, they will develop into two main phases, which is the early thymocyte development and also selection event. Okay, so during your thymocyte event development, your immature thymocyte, which is your CD4 negative and CD8 negative, and pass through the serval stage DN1 until DN4, during which they commit to the T cell lineage and begin to rearrange their T cell receptor. So very general, this stage is the formation of T cell receptor. They need to produce the T cell receptor. And then to express the protein. So they are the two protein we call as CD4 and CD8. One of the reasons why we say T4 and T8, helper T cell and also cytotoxic T cell. That's one of the reasons. One of the reasons why we say T4 and T8. T4 because it expresses CD4 protein. T8 because it expresses CD8 protein. But now, during thermocyte, knows this protein as well. So this process is to express this protein and also to produce the T cell receptor. Okay, after that, they will produce positive CD4 and CD8. So the cells now containing CD4 protein and also CD8 protein now. Okay. Next, your DP thymocyte, DP thymocyte. Okay, so double positive lah. I say this is double positive thymocyte, meaning that what is recalled as double positive. This one is double negative. Double negative meaning that there are no express. The T cell does not express any protein yet on their surface, and then after that they will express the protein, but both protein will be expressed. CD4 and CD8 are on the cell surface. Okay. And next, after that they will go through the selection. They will only select self-MHC and self-peptide combination with their TCRS. So double positive thermocyte whose receptor bind to MHC peptide with intermediate affinity are positively selected and measure to single positive T lymphocyte. Okay, so this positively selected thymocyte continue to mature and migrate to the medulla, where they are subject to another round of negative selection to self-antigen that include tissue-specific protein. So there are two processes at here. The first process is positive selection, and second process is negative selection. So meaning, what are their meaning? Okay, so this data positive, the first selection, positively selection, you need to select which protein. They only want to express the one protein only, either CD4, either CD8. So throughout this positive selection, when they bind to this cell MHC complex, so they will know this cell only can express CD4. This T cell only can express CD8. Okay, once they express, I already mentioned before, just now, you need to know that our T cell, the T cell receptor is only want to attack harmful antigen. They don't want to attack self-antigen. They don't want to attack our body one. So they need to test. They need to select only the cell, only the tissue, only the antigen. Ensure that the T cell can attack the harmful antigen only. So that is why that you call as negative selection. Because if positive selection meaning that it can attack self-antigen. So we don't want that. So if there are positive selection occur, so this cell will be later will be die. It will denature. But the negative selection later it will go lah. So finally, your major T lymphocyte will be developed and with CD4 protein and CD8 protein. So now your cell is T cell express CD4 is known as helper T cell, or another name T4 cell. The cell T cell express CD8 protein is known as cytotoxic T cell, or we call as T8 cell. And then these two cells, please remember it, you need to keep in your mind, because these two cells are very important in your adaptive immune response. Okay. So actually, this is the overall of your development of T cell. So you can see, right? So after that, this is the, and your bone marrow, right? I already mentioned your T lymphocyte will be produced in the bone marrow. Later it will go to the thymus. Thymus is the place to your maturation of your T lymphocyte. So after that, they will go to process, right? Early thermocyte development, and also selection process. So in the development, the final product is to produce the T cell lymphocyte, they containing the T cell receptor and the double positive protein. And then after that, they will go through the positive selection and negative selection, and finally produce the cytotoxic T cell and also helper T cell. Okay, so that's all about the development of T cell. Yeah. Another one is development of B cell. Okay. About the development of B cell, you need to know more easy compared to the T cell, I can say. Yeah. So in development of B cell, I just use diagram lah. So we know that B cell are produced in bone marrow and developed in bone marrow, right? Mature in bone marrow too. So B cell, the most important thing is their receptor. Because it only containing one type of B cell. Compared to T cell, you need to have a first selection first, because you need to select which type of T cell first. You need to select either it is helper T cell or either is cytotoxic T cell. But B cell know this problem directly. So the B cell, only the problem is the B cell receptor, which we call as antibody. Yeah, antibody. Because B cell is the place to secrete antibody. So when the antibody still bind to the B cell, we call as B cell receptor. They need to select the antibody. So this is the general stage of your B cell. So first stage, they will go to the pro B stage, and then later it will go to BB stage. So starting this stage, B cell receptors start to produce, but still immature. But still the receptor still immature yet, does not have the specific yet. But for now, immature B cell is starting to secrete, which they already containing the B cell receptor. Which IgM already. IgM. After that, it will become mature, where it will secrete IgD. So more easy lah. Yeah. So the overall process of your B cell development. So this is the explanation lah. Okay. So yeah, clear about your B cell. Okay, now we go to antibody. So I already mentioned that B cell is a place to produce your antibody. So when it is still adhere on your B cell, we call as B cell receptor. So you can see here, see that diagram. So antibody is the Y-shaped protein. We know that antibody is one type of protein, right? One type of protein. Okay, so it is secreted from B cell and it is a Y-shaped protein. You can see this. And it is synthesized from B cell and consists 4 polypeptide chains. So it is one type of quaternary protein too. Yeah, because it contains 4 polypeptide chains. So you can see these 4 polypeptide. Why contain 4 polypeptide? 2 heavy chain and 2 light chain. So yeah, 1, 2, 3, and 4. So they are containing 2 heavy chain and also light chain. So light chain is the chain at the upper side one. Heavy chain is the chain that will bind toward the B cell surface. If we that provide Y shape lah. Senang cerita. Heavy chain is the place that provide Y shape punya. And light chain is the place that bind to the heavy chain. And then each chain containing constant and variable region. So you can see got C and V, right? C is constant region, V is variable region. So you will see this region already mentioned. C region is where your amino acid sequence are changed very little, almost same. All the antibody for this sequence, we call a constant region. At this region, all your amino acid sequence are almost changed, 99%. Just only got very little change only. Maybe got 1, 2 to 3 amino acid are different, but variable change are totally different. And that is why that your antigen binding site is at here. Antigen binding site is the antigen will bind at here. Antigen will bind at here. They will not bind at here. They will not bind at here. Only will bind at here. So that is why that your variable region is at the upper part. Your antigen only can bind at here. So your variable chain are totally differ from every type of antibody. Every type of antibody will have different variable region. The sequence or amino acid are totally different. So variable region consists of antigen binding site, where it is the place your antibody bind to specific epitope of your antigen, and then the regions are bonded by disulfide bridge. So you can see the constant region, right? The constant region, the heavy chain and light chain are bonded by the disulfide bridge. Okay. For this one, I need to tell you about the type of antibody. So there are total 5 major type of antibody. So why got 5? And then you see the name MDGAE. Why not say ABDCE? Muy jili macam ni susah hafal tau. MDEAG. Aloy. Kenapa tak mak ABCDE Senang kan ABCDE, more senang. Kenapa nak ni because it depends structure of their change. Depend on the heavy chain. Based on discrete terms. We know that this one is gamma, right? Gamma, mu, alpha, epitope, episode, and then this one is delta. So that is why that gamma, right? IgG, and then this one is mu, IgM. This one is alpha, right? A, IgA, and this one is episode, E, IgE, and then this one is delta, IgD. That's why we name this as like this: IgG, IgM, IgA, IgE better than A, B, C, D, E. That is why one of the reason. Because the scientists we found this heavy chain, so they named it based on the first alphabet So gamma, right? Gamma, G, so IgG. Mu, right? Mu, M, MU, so IgM I, alpha, A, IgA episode, E, IgE delta, D, donkey, so IgD. So that is why that they naming like this. So okay, so okay, so there are in the scientists that discover there are total got 5 heavy chain only. They don't show this kind of structure: gamma structure, mu structure, alpha structure, episode structure, and also delta structure. So that is why that we containing 5 class of antibody only. So they classify this. And then light chain, or how about light chain? So light chain only got 2 type, either kappa or lambda, but both of them be able to occur at any way. So you cannot classify based on these. So we only can classify based on this heavy chain. Okay. I just want to tell you. And another one is a J chain at here. So this chain show that the structure of the antibody. So you can see only IgM and IgA containing J chain. Others does not have. Meaning that J chain, meaning that if none have J chain, meaning that the antibody structure is like this. We recall as monomer structure. Only like this. They show this kind of structure only. But for IgM and IgA, because due to the J chain, so there are many antibody. The antibody, this antibody structure are gathered together and join become J chain. So I give you the diagram. You can see this. If you can see this, there are IgN. You can see there are 5 antibody, right? 1, 2, 3, 4, 5. There are 5 antibody here. So they are combined to each other. They are bind to each other through the disulfide bond, because disulfide bond, and also J chain at here. So there are J chain at here. So J chain is play the role to bind between the amino acid. And another one is IgA. If you see this now, they are also the J chain. So the IgA is dimer structure, and then IgM is pentamer structure, because they're containing 5 B cell receptor at here at the same time. So this is the one antibody. We cannot say that IgM containing 5 antibody. No, IgM structure is memang like this. They will become like this structure we call as pentamer structure. So what are the function of IgM? So it is the first antibody produced in the humoral immune response. So what is humoral immune response? This one later I will explain it. But you need to know antibody is produced from B cell, right? So you need to know that if the IgM is produced, so when you get infection, your first antibody produced is IgM. Remember this. IgM is the first antibody antibody produced when you get infection. Need to know. Okay. Another one, IgD. Okay, IgD actually is the I can say is a useless function, because for now, until now, scientists do not discover any specific function for this antibody. They only found that this antibody play the role in maturation of B cell that help activate B cell following antigen binding. They only found this function only. There's no function anymore. So this B cell, the IgD normally is help to activate B cell only lah. And this is monomer structure. Okay, another one, IgG gamma. Okay, IgG. So IgG, most complicated. It is the only antibody can be transferred through the placenta that protect your baby, protect fetus. So this is it is only the antibody can be produced to transport this antibody to protect your fetus. Only this. Others antibody cannot. IgD or even IgD also. IgD is the first antibody, but this IgD cannot. Not IgD. IgM. Sorry. IgM. IgM cannot across the placenta one. They cannot protect the fetus one. Only IgG. They found that. So another one, IgG is dominant in secondary response too. Meaning that when you get first infection, same disease, same A disease, already get this disease, A disease, and then maybe few months, maybe one later, you get the A disease at the same time. So for now, at that time, your immune response will not secrete IgM anymore. IgM anymore will not secrete IgM. Not say that not secrete anymore. The secretion of IgM will be decreased, but it will secrete more IgG because it is dominant in secondary response. IgG will be in large amount compared to IgM. Okay, so IgG also is the monomer structure. It only containing a monomer structure. Okay. Another IgA, another antibody that containing J chain, which is the dimer structure. You can see this is a dimer structure. So the IgA is the antibody that need to prevent virus and bacteria from attaching to epithelial surface. So this antibody play the role. Normally you can find it in mucus, the place that can secrete mucus, like your nasal cavity, your epithelial cell, gastrointestinal gut, digestive gut, gallbladder. All these that can secrete mucus or in the epithelial surface like kidney too. IgA will be produced. IgA play the role. It protect them. So it is a dimer structure, another spatial structure. Another one, final one is IgE. Okay, this antibody play the role in allergic, inflammation, and also parasite infection. Parasite worm. So if got parasite worm or the big pathogen, especially big pathogen like fungi or the huge bacteria, or you have allergy, respond to allergy. So it can bind to the mast cell. So mast cell is one type of leukocyte which containing histamine that trigger many allergic symptoms. So it is to activate mast cell. So the IgE will bind to mast cell, so that it activate mast cell, so it will trigger this allergic reaction. So normally it is also immediate in hypersensitivity reaction that are responsible for the symptom of hay fever, asthma, high, and also anaphylactic shock. So this is the monomer structure. So the most important thing is inflammation, allergic reaction, parasite infection, or the huge bacteria or fungi like that. IgE will take response. And also another function is about the high temperature. It will respond in high temperature too. So this is all about IgE. Okay, so that's all about the first topic. So the first topic is only the general overall. Now I go to the immunity development that I say before, like activation of B cell, activation of T cell, and how the adaptive immune response, how your body fight against, and all the mechanism about the third line defense. Yeah, occur at they will learn at here. So far any question ma? No question. Okay, now we go to the development immunity, which is the most important part, where the in examination will most coming out in this part. Okay, so in this topic you will learn about three things. You need to learn about activation of adaptive immunity. Second, cell-mediated immune response. Another one, humoral immune response. These three things, and then these three things are related to each other one. So the most important is cell-mediated immune response and also humoral immune response. These two, if you want to activate it, if you want to trigger these two response, you need to have adaptive immune response activation first. You need to activate first. If not, that two will not trigger. So how to activate? So as I mentioned, antigen presenting cells. So this is the general about the diagram. So yeah, the most important thing I need to show you is antigen presenting cell. This one. This is very important because antigen presenting cell play the role to activate your T cell, because you need to know whatever T cell, CD4 or CD8 T cell, helper T cell or cytotoxic T cell, any T cell, they are not activated yet. So they don't know which kind of antigen they need to attack. They don't know, so they need to realize on antigen presenting cell. Okay, so if you see when the antigen presenting cell starting to bind to them and they activate, and then all this process. So in our syllabus, you need to load all these process. Based on this diagram, you know. For me, I know that because I already know. So now I need to teach you how to use this. Okay. So the first process is upper step first. You need to have this helper T cell. So what is we call as helper? Why we naming as helper? Because he help to activate cell-mediated immune response and also humoral immune response. He need to help first. If no, then no adaptive immune response. Okay. First, helper T cell. Helper T cell play the first role first. Either you want to trigger adaptive, again, if you want to trigger cell-mediated and also humoral, both of them first, you need to activate helper T cell first. You need to activate helper T cell first. If no helper T cell, then no cell-mediated and no humoral. You need to know this. Okay. Helper T cell. Okay. Helper T cell play the role in activation of adaptive immunity. So helper T cell is the effector cell of T cell that consists of CD4 protein that activate humoral and cell-mediated immune response. Yeah, very important. Okay. Before that, you need to activate first. You need to activate helper T cell. There are another process. Before that, you need to have antigen presenting cell first. You need to antigen presenting cell. So what is antigen presenting cells? So antigen presenting cell actually is macrophage or dendritic cell. So in our syllabus, you just know about macrophage first. Okay, macrophage. Macrophage can play the role in antigen presenting cell. So if you still remember in SPM, recap again in your SPM, phagocytosis. Okay, you now learn about that. Macrophage play the role in second defense system, and it play the role to undergo phagocytosis to engulf bacteria. By one of the function. Another function of the macrophage in adaptive immune response, it can act as antigen presenting cell. So if you see this term, antigen presenting, so meaning that it will present the antigen, show the antigen at the surface of the cell, like this. We can see. So meaning that this macrophage, it will engulf your bacteria, engulf your pathogen, engulf, semua makan, lysis, all this, it will produce antigen. And then this antigen will bind with the one component we call as MHC2 class 2 molecule. Okay, what is MHC? I will explain it later in the topic concept self and non-self. But for now, you need to know in macrophage they will undergo the phagocytosis process and then lyse the bacteria to produce antigen. Okay, so this antigen will bind with MHC2. MHC2, so there are two types of MHC, 1 and 2. Only bind 2. You cannot say bind to MHC1. If you say bind to MHC1, then totally wrong. MHC2. Okay, that later it will move toward the cell and then it will present this MHC complex. We know as MHC complex now. Because the MHC now is already present your antigen at here. So now your macrophage is antigen presenting cell, short form APC. Okay, so later I will use APC. Okay, next. Class 2 molecule provide a molecular signal which your APC is recognized. Okay, APC will display the antigen fragment complex compared with the class 2 MHC molecule on the cell surface. So now your APC will display all the antigen fragment complex with class 2 MHC, so to MHC. So your single antigen receptor, so your helper T cell will containing the receptor, T cell receptor. That's why I need to say antigen T cell receptor. So that is why during the development of T cell, you only can choose the cell, the body only can choose the T cell receptor that can only attack the harmful antigen only. If this T cell receptor can recognize self antigen, then it will attack the self antigen, our body cell. So T cell receptor is very important too. So this T cell receptor will bind to the bind to this complex, bind to the antigen fragment and the Cas2 MHC molecule. So you bind to it, and another one at the same time there are also CD4 protein. This CD4 protein will bind to the Cas2 MHC2. Okay, so this CD4 protein will bind to the Cas2 MHC to keep the cell joined together. Joined together. After that, at the same time, your APC will release cytokine. Normally is IL-1 to activate your helper T cell. So you need to know there are three signal. First signal is your T cell receptor need to bind to the antigen. Another receptor is your another receptor is your CD4 accessory protein. CD4 protein will bind to Cas2, MHC Cas2. And then another receptor, another signal is your cytokine. We were chemical, you can say like a chemical substance that activate your by the APC to activate your helper T cell. So you need to these three signals together, then you can activate your helper T cell. Lose any one signal, helper T cell cannot activate. So you need to alert three signal. First signal, antigen receptor bind to antigen bound on the MHC complex, and then another one signal, CD4 protein bind to MHC Cas2, and another signal is APC will secrete chemokine, release chemokine to activate helper T cell. Three signal. Okay, now your helper T cell is now activated. Okay, once your helper T cell is activated, they will also start to release another cytokine we call as IL-2. The others now is IL-1. Now IL-2 for proliferation, meaning that it was starting to copy. Proliferation is copy and paste. It was starting to copy many activated helper T cell. So this helper T cell is only activated, only can recognize this antigen only. A antigen, we say as A antigen. So or A antigen. So all these helper T cell activated only can recognize A antigen only. So it already say helper, right? So this helper T cell will give the signal to the B cell and also cytotoxic T cell. So this cell will secrete the cytokine IL-2, help to activate and stimulate B cell for humoral immune response and cytotoxic T cell for cell-mediated immune response. Yeah, clear. First, you need to activate helper T cell. So when your helper T cell activate, it will starting to proliferate. At the same time, it will start to activate B cell and also activate cytotoxic T cell. So when the B cell receive the signal, then it will trigger humoral immune response. When your cytotoxic T cell receive the signal from the activated helper T cell, it will trigger cell-mediated immune response. Okay, clear. I hope clear lah. Okay. Okay, next is about cell-mediated immune response. Okay. So this is one first start. So these two are different. One is B cell, one is cytotoxic T cell, but the similarity is they need the signal from the helper T cell. Activated helper T, not helper T cell, activated helper T cell. You need to use the specific terms. Okay. Okay, clear. Okay, now I go to the cell-mediated immune response, which is the cytotoxic T cell first. Okay. Okay, now we go to cell-mediated immune response. Okay. This one is only involve cytotoxic T cell only. So you see cell-mediated, meaning that it will directly go toward the cell. So it only involves cytotoxic T cell to destroy the infected, not injected, infected host cell. Infected, not injected. Okay, meaning that it would directly kill the host cell that already infect. Like if your cell, like this cell, like this cell is already infected by the virus or already infected by bacteria, all that already infected, so this cell is already useless, unused anymore, so cytotoxic T cell will destroy it. So you need to know the mechanism how the cytotoxic T cell destroy this cell. Macam mana? Okay. So you need to activate first. You need to activate cytotoxic T cell. So how to activate? It receive the signal. Bila it receive the signal, dia dah dapatkan signal. Okay, dapat signal daripada cytotoxic T cell. Okay, dia dah tahu. It will only attack the cell infected, that cell infected by A antigen. If the activated helper T cell recognize A antigen, so this cytotoxic T cell only can attack, only can destroy the cell that affected by A antigen only. If it is B antigen, this cytotoxic T cell cannot attack. It only can attack the cells, the host cell that infected by A antigen only. Highly specific. Okay. So this activation requires signal for activated helper T cell and also interaction with antigen presenting cell. So this antigen presenting cell is the host cell. So similar what you see in antigen in macrophage, your host cell actually also when once the infected by the antigen, by the pathogen, they will also display the antigen too. But now your MHC complex is not class 2, is class 1. 1. Just now activated helper T cell is class 2, right? Your macrophage is present in class 2. Now is class 1. Why? One of the reason is all the host cell in our body, whole body cell will express class 1 MHC molecule. It will express class 1 MHC molecule. Only certain cell express class 2. Macrophage. Only macrophage. Macrophage and also dendritic cell, they will express class 2, and also another one is B cell. Later we will see it. Okay, I just want to show you that all the host cell will express class 1. So that is why that the antigen will bind to class 1 to form the antigen class 1 complex. Okay, so once they already present antigen or cytotoxic T cell akan jalan. Already you see signal. Okay, dah tahu lah. Okay. Dia jalan. Okay, saya nampak. Okay, nampak. Saya tengok. Ini sel ni ada A antigen ni. Yang saya kena tangkap. Ini yang saya kena tangkap. Okay, dah bind dekat sini. You use the antigen receptor bind to the use its antigen receptor bind to the antigen on the class 1 molecule, and then another protein, CD8 protein will bind to the MHC1 so that recognition of the cytotoxicity will occur. So so join the cell together. So another one, so when it is start join together, already recognized, okay, your cytotoxic T cell will start to release perforin. So this is one type of enzyme, perforin, not enzyme, to create the pore in the infected cell part of your plasma membrane. So that it create the pore, so that all the water molecule will rush into the cell, resulting in lysis process. And also it will produce another enzyme called as enzyme that break down the antigen and enter the infected cell by endocytosis. So the enzyme will initiate apoptosis within the infected cell, resulting in fragmentation of your nucleus and cytoplasm, and finally die. So the key point is it will kill the cell. So in general, you want to trigger cell-mediated immune response, only cytotoxic T cell involved. And then this cytotoxic T cell need to activate first. It receive the signal from the activated helper T cell. Once it receives signal, it will go around cari tempat. Okay, nampak lah ini cell dia ada A antigen ni. So it will bind together and then release perforin and enzyme to kill this cell. Okay, simple right? Man, sangat easy. Okay, yang ni another diagram lah. Okay, next I go to humoral immune response. Okay, humoral immune response. Humoral immune response involve only B cell. It will only involve B cell only. B cell only. Cytotoxic T cell is only special for cell-mediated immune response. Humoral immune response is only involve B cell. Okay, so similar to what in cytotoxic T cell, it also require the signal from the activated helper T cell. But at the same time, your B cell is also act as the antigen presenting cell too. So at the same time, your B cell is also need to engulf, take the pathogen. So if you want to activate the T cell by the helper T cell and also binding of B cell with the antigen which have the same epitope with the helper T cell. Yeah, meaning that when your this helper T cell bind recognize A antigen, meaning that this B cell not activated yet, also need to find a similar antigen. So dia akan cari juga lah, cari dia macam cari, dia tak buat kerja kan, tak buat kerja, so dia tahu kerja dia engulf antigen je, makan antigen je. Okay, dia cari, ada antigen makan, ada antigen makan semua, tapi makan semua. Okay, once it engulf it will also act as APC too. Display the antigen fragment with class 2 MHC molecule. Also class 2, not class 1. Just now, cytotoxic T cell is class 1. This one is class 2. Similar what you see in macrophage. So, apa yang berlaku dekat macrophage, awak just copy and paste, berlaku dekat B cell je. Just that, it will bind to with class 2 MHC compact on the cell surface. So, your activated helper T cell will bind to the antigen fragment and class 2 MHC of your B cell. So, yeah, faham? Okay, faham kan? Okay, now your B cell is already activated. So, once your B cell is starting to activate, it will start to secrete two types of B cell. One is plasma cell and another one is memory cell. Okay, so, plasma cell and also memory B cell. Okay, so in general, I just focus on plasma cell first. So, your plasma cell now was starting to secrete antibody. This plasma cell is immediate response. When it go to plasma cell, so dia akan pergi tempat. If there are infection occur, then this plasma cell was starting to release the antibody. It was starting to secrete antibody, and then this antibody are specific to that antigen, and that antigen only. So, antibody can be transported by lymph and also blood to the infected region lah. Okay. So, memory B cell, not only B cell containing memory, any cell containing memory too. So, if you see this diagram, refer this diagram. So, you see helper T cell also have memory, B cell also have memory, cytotoxic T cell also memory. So, anyway, I just want to tell you that once all these cells activated, helper T cell, cytotoxic T cell, and B cell are activated, they will also proliferate, become memory cell. So, what are the difference? So, the main difference is, if this is helper T cell, it will activated helper T cell, it will directly provide response, immediately. But if it is memory, stay at here, dekat sana je. Dia macam sleeping je lah. Dia is already activated. The helper T cell are already activated. Just that dia tidur je. Dia pergi surrounding je. Dia tak buat. It will not provide the signal, provide the chemokine to the B cell and also cytotoxic T cell. Dia just stay at there. And then once the first infection already done, or already semua dah done, so this memory T cell akan pergi tempat-tempat lah, akan pergi sini, pergi sana. Maybe it will go to your stomach, maybe it will go to your heart, maybe it will go to the brain. And then once you get the second infection, when you get the second infection with the same antigen, like this activated helper T cell recognize A antigen. So when you get the second infection, again with the same antigen, A antigen again, this memory T cell will directly activate. It will directly activate to become helper T cell and directly trigger immune response. So it does not require again antigen presenting cell. tak perlu lah. Dah tak perlu ini antigen presenting cell lah. Dah tak perlu. Directly pergi. Directly. Faham? Ini beza dia. Directly. So, you don't need to take time. Once there are the second infection, this memory T cell directly activate and directly give the signal to B cell and cytotoxic cell. So, it will trigger the immune response. If in the traditional pathway, you need to have antigen presenting cell first, macrophage first, because macrophage trigger phagocytosis is during second defense. You get defense. Yeah, so it take time. That kena guna masa. So it's right that why some of the patient in COVID-19 can get well soon and some of them cannot get well because of this immune response. Because this third line defect, it require time. It does not immediate respond. It require time because the hepatitis cell need time to recognize it. So some of the patient, they cannot wait. The virus already infect your lung already infect the large area of your lung. It already cannot wait until your hepatitis cell recognize it. If the wait until hepatitis cell recognize it, the patient already, that's how you can say, already near to die lah, and even die. So that is why we got memory hepatitis cell. That is why the people that get second infection, although you are not getting any vaccine, but you can still get well soon compared to the first time that they will get COVID-19, because you already got memory as well. Okay, so similar to your memory B cell and also memory cytotoxic cell, they also stay as memory. So once there are the second infection with the same antigen, they will directly trigger and directly provide the response. So this is why we call as memory. So that is why very many people say that when you get the first infection with the same disease not say disease, kalau awak dengar lah orang tua tu, kalau awak dah dapat penyakit ni, the next time awak tak akan dapat, or even awak dapat cepat sembuh, but actually not very accurate lah, but the concept is true because based on this, we containing memory. That is why that when you even go to get the second exposure, second infection within the same antigen, you still the adaptive immune response can be respond in very fast time. Okay, so I think we stop until here. So any questions so far? I think no question as well. Okay, so I hope that today topic can let you guys more understanding lah about the immune response, and then this immunity development are very important topic, and many it will come out in SA1. Come out even when you want to come out this chapter, normally possibility it will come out this topic. So yeah, you need to alert lah. Okay, so I think that's all for today. So I will end the class now. So for tomorrow, I will finish up the end topic of this chapter lah. So possibility next week we will finish up the Sem 2 syllabus. Okay. So if no question, then we dismiss our class now. So thanks for your joining and have a good rest. Good night. Bye.